Hello everyone, welcome to India's largest online learning platform ITLS Academy. The topic of today's discussion is the adult stem cell. We have discussed a brief overview and an brief introduction about the adult stem cells in our part 1. This is part 2 and uh, we will see a detailed study of adult stem cells in this particular lecture. We were studying the plasticity of adult stem cell in our previous lecture. In today's lecture, we will start with experimental evidence of adult stem cells and plasticity. What is plasticity? We have studied in our previous lecture. If you haven't watched, then I recommend you to watch that particular lecture first and then start this particular video. So for the experimental evidence, first of all we will see adult stem cells of the nervous system. In this lecture, we will focus on the adult stem cells of nervous system. So let us see an introduction. First point, more than 30 years ago, Altman and Das showed that two regions of the postnatal rat brain, the hippocampus and the olfactory bulb, these two regions are the postnatal rat brain, and these contain dividing cells that become neuron. Despite this, the prevailing view at the time was that nerve cells in the adult brain do not divide. Next point is, today scientists believe that stem cells in the fetal and adult brain divide and give rise to more stem cells or to several types of precursor cells. Here in this diagram you can see this is a neural stem cell. This neural stem cell has the property of self-renewal, that it can give rise to a cell that have the same shape and size and function, that is self-renewing. Apart from this self-renewal property, these neural stem cells also differentiate into many types of cells, like these may be neurons, these may be astrocytes, and also these may be oligodendrocytes. Neuronal precursors, also called neuroblasts, divide and give rise to nerve cells. So we can say that nerve cells or neurons are derived from neuronal precursors, of which there are many types. Glial precursor cells give rise to astrocytes or oligodendrocytes. Except these glial precursor cells, astrocytes are a kind of glial cells. These are a type of glial cells which lend both mechanical and metabolic support for neurons. They make up 70 to 80% of the cells of the adult brain. Oligodendrocytes make myelin, that is, a fatty material that enshapes nerve cells, axons, and speeds nerve transmission. In contrast, a fetal or adult CNS, that is central nervous system, that contains the brain and spinal cord. So fetal or adult CNS stem cells may give rise to neurons, astrocytes or oligodendrocytes that you can also see in this diagram. And these differentiated cells will depend on the signal it receives and its three-dimensional environment within the brain tissue. Here in this diagram you can see this is neural stem cell. This is formed by isolation of primary CNS tissue, that is spinal cord tissue, adult brain or fetal brain. This all can give rise to neural stem cell. Despite this, differentiation of pluripotent stem cells. These pluripotent stem cells differentiate and give rise to neural stem cells. Next is transdifferentiation from somatic cells. These somatic cells may be chemical compounds, growth factors, and transcription factors. All these factors from the process of transdifferentiation can give rise to neural stem cell. If we talk about the therapy of neural stem cell, then first one is paracrine effects and next one is cell replacement. In the paracrine effects, secretion of soluble factors. These soluble factors may be neurotropic factors, cytokines, and growth factors. If we talk about the cell replacement, here you can see differentiation of neural stem cells. Neural precursors forms neuron. Oligodendrocyte precursors form oligodendrocytes. And the glial precursors form astrocyte. This is the process of cell replacement. One cell is replaced with another cell. Central nervous system stem cells in the subventricular zone. Very first point, central nervous system stem cells found in the forebrain that surrounds the lateral ventricles and are heterogeneous and can be distinguished morphologically. Ependymal cells, which are ciliated, line the ventricles. Some of these cells divide rapidly while others divide slowly. Next point. A recent report indicates that the astrocytes that occur in the subventricular zone of the rodent brain act as neural stem cells. The in vitro assay to demonstrate that these astrocytes are in fact stem cells involve their ability to form neurospheres, and this is the grouping of undifferentiated cells that can be dissociated and coaxed to differentiate into neurons or glial cells. Next point, as long as mitogen is present in the culture medium, the cells continue forming neurosphere without differentiating. To induce differentiation into neuron or glia, cells are dissociated from the neurospheres and grown on an adherent surface in serum-free medium that contains specific growth factors. So now we will see central nervous system stem cells in the ventricular zone. The point is, another group of potential central nervous system stem cells in the adult rodent brain may consist of the ependymal cells themselves. Next point, ependymal cells, which are ciliated, line the lateral ventricles. They have been described as non-dividing cells that function as part of the blood-brain barrier. However, in a recent study in which two molecular tags, that is, the fluorescent marker DIL and adenovirus vector carrying LAC-Z tags, were used to label the ependymal cells that line the entire CNS ventricular system of adult rats. It was shown that these cells could indeed act as stem cells. Next point. Collectively, the data suggest that CNS ependymal cells in adult rodents can function as stem cells. The cells can self-renew and most proliferate via asymmetrical division. Many of the CNS ependymal cells are not actively dividing, that is, in the quiescent stage, but they can be stimulated to do so in vitro, and this can be done with mitogenes or in vivo in response to any injury. After injury, the ependymal cells in the spinal cord only give rise to astrocytes, not to neurons. Central nervous system stem cells in the hippocampus. Hippocampus is a part of the brain. First point, the hippocampus is one of the oldest parts of the cerebral cortex in evolutionary terms and is thought to play an important role in certain forms of memory. The region of the hippocampus in which stem cells apparently exist in mouse and human brains is the subgranular zone of the dentate gyrus. In the human dentate gyrus, some brdu-labeled cells express NEU-N, that is neuron-specific enolase or calbindin, all of which are neuronal markers. The labeled neuron-like cells resemble dentate gyrus granule cells in terms of their morphology as they did in mice. Other BRDU-labeled cells express glial fibrillary acidic protein, in short GFAP, an astrocyte marker. Next point is the greatest number of BRDU-labeled cells were identified in the oldest patient, suggesting that new neuron formation in the hippocampus can continue late in life. Here in this diagram, you can see adult hippocampus. This is activated RGL. This activated RGL can give rise to new RGL, new intermediate precursor, and new astroglia. New intermediate precursors further can give rise to neurons, and also these are self-renewing cells. These intermediate precursors can self-renew. I hope you all understood what is self-renewal because we are studying self-renewal since our last lectures. Apart from this activated RGL, this cannot give rise to new oligodendrocytes, although it can give rise to new astroglia but not to new oligodendrocyte. This activated RGL forms new astroglia and can form quiescent radial glia-like cells, that is RGL. Here you can see in this diagram, this is embryonic stem cell. This embryonic stem cell can form embryonic and adult neural stem cells. These embryonic and adult neural stem cells further can give rise to mature nerve cells. Here, these embryonic stem cells forms adult neural stem cells by neural identity genes. Further, these embryonic and adult neural stem cells give rise to mature nerve cells by the process of neural differentiation genes. Here you can see that neural identity genes and neural differentiation genes, two types of genes are involved. These two genes are not same. These have two type of functions at two different sites. Here this mature embryonic and adult neural stem cells gives rise to mature nerve cell. Apart from this, it can give rise to novel targets for pharmaceutical research and development in direct cell production, and also to unlimited cell source for transplantation. Both of these can be used in the process of brain repair. These can play an important role in the process of repairing of brain. Here in this diagram you can see how these stem cells are formed. This is zygote, a totipotent stem cell. When this zygote differentiates, it gives rise to blastocyst. After many cleavage divisions, a mass of cell form and this is known as blastocyst. This blastocyst have an outer covering known as trophoblast layer and a mass of cells at one edge known as inner cell mass. This inner cell mass further gives rise to pluripotent stem cell. After the culture of this pluripotent stem cell, this gives rise to embryonic stem cells. Also, this pluripotent stem cell gives rise to pluripotent somatic stem cell. This pluripotent somatic stem cell further can give rise to central nervous system, peripheral nervous system, hematopoietic cells, liver, skin, mesenchymal cells, etc. A lot of many more cells can be generated by these pluripotent somatic stem cells. These all cells generated by the pluripotent somatic stem cell are multipotent stem cells because these can further give rise to a lot of types of cells. Here, this blastocyst outer layer, that is trophoblast or embryo or gonads, after the culture, these forms primitive germ cells or germ-line stem cells. These germ-line stem cells further can give rise to pluripotent somatic stem cells, and in turn these pluripotent somatic stem cells can also give rise to these multipotent stem cells like CNS, PNS, hematopoietic system, liver, etc. So thank you guys for watching this session. This was all about the adult neural stem cells. We have almost covered this topic. Left out things we will study in our next video. So till now, guys, follow ITLS Academy on various social media platforms like Insta, Facebook, Twitter, YouTube, WhatsApp and LinkedIn. For latest videos and updates, subscribe ITLS Academy on YouTube and also press the bell icon. And you can also get the app of ITLS Academy from Google Play Store. So thank you very much guys for watching. If you like this video, then like it, share with your friends and also subscribe our channel ITLS Academy. Thank you very much again for watching this session. Stay connected with ITLS Academy, learn online, stay safe.